Welcome and thank you for standing by. At this time, all participants are in listen-only mode. During the Q&A session, if you'd like to ask a question, you may press star one on your phone. Today's call is being recorded. If you have any objections, you may disconnect at this time. I'll turn the call over to Ms. Irene Ahir. You may begin.
Thank you. Hello, I am Irene Ahir of CDRH's Office of Communication and Education. Welcome to the FDA's 47th in a series of virtual town hall meetings to help answer technical questions about the development and validation of tests for SARS-CoV-2 during the public health emergency. Today, Toby Lowe, Associate Director of the Office of In Vitro Diagnostics and Radiological Health, and Dr. Timothy Stenzel, Director of the Office of In Vitro Diagnostics and Radiological Health in the Office of Product Evaluation and Quality, both from CDRH, will provide a brief update. Following opening remarks, we will open the line for your questions related to the development and validation of tests for SARS-CoV-2. Please remember that during this town hall, we are not able to respond to questions about specific submissions that might be under review. Now, I give you Toby.
Thanks, Irene. Thanks, everyone, for joining today. I have a couple updates, and then I will hand it over to Tim for some more. First, I wanted to make sure everyone was aware that just this morning we updated templates on our website. We updated the serology template for test developers, and in that update we added some information about viral mutations and variants, and also did some clarifying edits throughout. So hopefully that will be useful. And then we also posted a new template for test developers of serology tests that detect or correlate to neutralizing antibodies. So I know we've had a lot of interest in that template and a lot of people waiting for it. So it has been posted, and we hope that it is also helpful for you. The other update that I have is that starting tomorrow, we will open up the ability for you to send questions by email ahead of time for us to consider prior to the town hall, so that we can try and address potentially more frequently asked questions. So there will be an email that goes out likely tomorrow with instructions on how to do that, and the email address will be cdrh@fda.hhs.gov. But again, you'll get an email. If you're on the town hall email list, you'll get an email with those instructions tomorrow once that opens up. And with that, I will turn over to Tim to give some additional updates.
Thank you, Toby, and welcome again today. I am hearing that some callers may be having trouble calling in, so if our technical team could check on that and assist those callers, that would be great. Let me start off with, prior to getting into the main topic, with a frequently asked question that we get about the use of frozen banked samples for validation of point-of-care tests, in particular antigen tests. And yes, we are allowing banked frozen samples to be used. And banked samples can be mixed in with fresh samples. We still are asking that at least 10% to 20% of the positives are composed of low positive samples, which reflect the natural distribution of SARS-CoV-2 viral loads. We have seen that even early in symptomatic disease that CTs can be relatively high. Per the antigen template, we do want to see these low positives, at least 10%. And then we do want to see fresh samples as well, a minimum of 5 positive samples pre-market. And this is because we have noted in multiple times now, both for molecular and for antigen, that freezing can actually, for still specifically determined reasons, allows a greater sensitivity. And so freeze-thaw studies can show that the performance is the same or different, but just to be safe, we do want to see a minimum of 5 fresh samples, pre-market positive samples, and then we would ask you to fulfill that, the rest of the samples being fresh after authorization. Okay, with that, I will move into the main topic. So yesterday, we issued a new policy on serial screening, and particularly screening asymptomatic individuals, which we view as a critical part of reducing the spread of COVID-19. Screening involves testing asymptomatic individuals who do not have known or suspected exposure to COVID-19 in order to make individual patient decisions, such as whether an individual should participate in an activity based on the test results. Yesterday, we also issued a fact sheet to assist schools, workplaces, communities, and others looking to establish testing programs to screen asymptomatic individuals as they are selecting a test for screening. We also posted a new template for molecular and antigen tests for serial screening. We expect that the recommendations and information we provided here for test developers will help streamline the path to an EUA authorization for screening tests. Specifically, the template outlines situations in which FDA may authorize certain tests for screening with serial testing prior to the developers conducting certain performance evaluations with asymptomatic individuals. This may include, in certain circumstances, authorizing a point-of-care and at-home tests for OTC non-prescription use, if all the appropriate studies have been, other studies for usability and user studies and user comprehension have been performed. In this situation, we are leveraging evidence of a test's strong performance in symptomatic patients, combined with serial testing to mitigate the risk of false results when testing asymptomatic individuals for the cases in which we have not reviewed the asymptomatic testing performance pre-market. We expect to see that in the post-market studies showing and demonstrating adequate performance in the post-market setting would demonstrate that adequate performance with the serial testing plan. I want to back up a little bit, talk about how important home tests, point-of-care tests, and in particular antigen tests are. So starting in January 2020, the FDA and other U.S. government entities reached out to companies that are capable of producing antigen point-of-care tests and ultimately, obviously, home tests, which we've authorized. It wasn't that long into the year that we authorized our first antigen test. And then to date, I think we've authorized 15 antigen tests. We are very interested in home use, and on July 29th, 2020, we issued a template with our recommendations for home validation so that we can authorize both prescription and over-the-counter testing. In September of 2020, in a piece in The Hill, we suggested that for tests that may be performing lower than recommended levels, could use serial testing to improve overall performance using a combined results of 2 or more tests to be able to achieve good level of sensitivity. To date, though, we've received no formal EUA submission of a fixed serial testing plan showing that it is functioning. We hope that this new pathway, this new policy, will accelerate the use of serial testing to reopen schools and reopen workplaces and other situations, and say to keep convalescent settings safe. We recently, the FDA was recently briefed on a very well-designed, well-executed study that used serial testing of EUA-authorized molecular test and an EUA-authorized antigen test. And although it clearly showed that on a head-to-head competition or comparison, the antigen testing was significantly less sensitive than the molecular test. It also demonstrated that a serial testing program for the antigen test significantly mitigated the risk of false negatives in that serial testing program. So that was sort of the first really firm, good evidence that we had and helped usher in this new policy. So throughout the year, last year, we've striven to provide flexibility and adaptability in approaches to validate all tests, including home tests, screening tests. So we have authorized a number of screening tests, I think almost a score, and we hope that this greatly accelerates that as well. I wanted to go into a few more details. CLIA requirements are overseen by CMS, and FDA's actions in this policy are not removing any CLIA requirements. Of course, the use of at-home tests for self-testing is not subject to CLIA requirements. This new template is also not automatically changing any authorizations. It is providing a streamlined path for test developers seeking serial screening claims. So in order for a test to be authorized for OTC or non-prescription use, such test needs to have previously shown asymptomatic screening data. But with this, in order to be an OTC test. So, but now we are allowing that data for asymptomatic performance using screening to be revealed to us after authorization. This template provides an option for screening authorization prior to collecting data or completing the validation for asymptomatic individuals. We would still expect validation in symptomatic individuals as well as usability, user comprehension, and layperson appropriate labeling for an OTC test. Where a developer has already submitted that information, they would not need to submit more for FDA to authorize their test for OTC, though. They can simply come in with a supplement and amendment application asking to convert to OTC based on the serial testing program. Of course, the older pathway still exists. If someone comes in with meeting the performance expectations for an OTC test on a one-time test performance, we will continue to authorize that without the recommendation for serial testing. And that may be still an attractive option. Obviously, we've already authorized many OTC collection kits and some OTC home tests. So, and of course, providers can still order tests that are authorized only for diagnostic purposes, and we're not going to object to the off-label prescription use of those tests that don't have a screening claim. The FDA does not determine what claims a test developer decides to request for their test or their application. We are encouraging them to pursue this option if it's applicable to them and they are interested in it. We do authorize tests for certain indications and for certain use in certain settings as appropriate by their validation. Generally, point-of-care tests are authorized for use in the settings under a CLIA certificate of waiver with healthcare workers that are untrained non-laboratorians. These tests typically include an instrument and require a trained user to operate them, but not always, and it's not required to have an instrument. CMS is responsible for oversight of CLIA certification, of course. Tests that are authorized for at-home use are appropriate for self-testing without a trained operator and may be used for self-testing at home or self-testing in other settings, such as schools, workplaces, and are not subject to CLIA requirements in that specific narrow self-test category. Going further, each authorization indicates whether a test is for prescription or non-prescription use. Or as we said before and reiterated in the fact sheet when we issued yesterday, test authorized for symptomatic claim can be ordered by a provider for screening purposes. We made that clear earlier in this discussion. Some entities may use a blanket prescription to cover screening tests for their entire population. And if a test is authorized for OTC, over-the-counter, no prescription, of course, is required. And as we've said previously, including a prior FAQ and reiterating the fact sheet we issued yesterday, clinicians can absolutely order tests for off-label use of screening. We're not going to object to that. It is important, however, to be aware that certain tests include labeling that state the tests should only be used for symptomatic individuals. These tests should not be used for screening, and they would this pathway would not be open to them. However, we do know that some groups are setting up testing programs for back to school, back to work, and prefer to use a test that is authorized for screening. That's an important point. We have definitely heard resistance about using tests off-label from some quarters and from some clinicians. And this hopefully paves the way for getting rid of that as an issue. And once we authorize a test for screening, they, of course, are covered by the PrEP Act coverage. So again, we hope that this streamlined path will lead to more test developers seeking and receiving a screening claim. And as we've indicated, and this is primarily for antigen tests that will be serially tested multiple times a week, we expect to see a minimum performance sensitivity for symptomatic testing of at least 80%, with 70% at the lower bound of the two-sided 95% confidence interval, which we believe offers additional flexibility in obtaining a screening claim and using and including an OTC, over-the-counter use. So with that, that was a lot of information. We would like to go ahead and open up this call for questions, and look forward to that. Thank you.
The phone lines are now open for questions. If you would like to ask a question over the phone, please press *1 and record your name. If you'd like to withdraw your question, press *2. First question in the queue is from Shannon Clark.
Before we take the first question, this is Toby Lowe. I just wanted to jump in with one additional point of clarification. Tim talked about healthcare providers ordering tests off-label, and since there was quite a bit of discussion about at-home testing in that, I just want to clarify that a healthcare provider can order a test for a different indication, but they cannot order a test for home use if it is not authorized for home use. And with that, I will turn it back over to you to take the first caller.
Yeah, I didn't cover that caveat. Thank you, Toby. Appreciate that.
Yeah.
First question in the queue is from Shannon Clark. Your line is open.
Good morning. This is Shannon Clark with UserWise Consulting. We specialize in human factors testing, but I have a question about limit of detection determination. So to determine a limit of detection for an antigen test kit, do you have any requirements regarding which strain of inactivated virus is used to create contrived samples in the calculation of limit of detection? Can we just use one U.S. strain in order to create the contrived samples and determine the limit of detection?
So, for molecular tests, I just explained on a RADx call earlier this morning, some of them are going to be looking at the use of degenerate primers to make sure that they're covering the various mutations that are in circulation if they cannot engineer the assay around, or they want to update their assay to maintain current levels of performance in the face of increasing numbers in locations and mutations. It could be that that in the use of degenerate primers there could be changes in LOD. So we're working through what that might look like. But for antigen tests, which I think you're probably asking about for antigen tests,
yes.
no known variant or mutation that could affect the performance of that antigen test. Of course, look to our mutation communication, a recent one, for how we're asking developers to start looking for potential issues with their antigen, molecular, or serology tests. But yes, at the moment, one strain can be used for LOD determination.
Thanks so much.
Next question in the queue is from Jeff Andrews. Your line is open.
Hi, Dr. Stenzel. Thanks for taking the call. I'd like to confirm that for an EUA antigen test that in symptomatics has a PPA above 80%, that you were saying that this new template that was released yesterday will permit authorization for a screening claim, and then in addition to the symptomatic claim. And I wanted to clarify whether that means that screening claim for asymptomatics is only with serial testing or would also include one-time use, and whether a post-market study is always required or whether that's a decision the FDA would make individually.
Yes, we're anticipating that the serial testing will bring performance in an unknown performance in an asymptomatic population up to a acceptable level. We will want to measure that per each device that test that's authorized in a post-market study to confirm that performance in the asymptomatic population reaches that 80% or more utilizing serial testing. Yes, the test authorized for, currently authorized for at least 80% sensitivity PPA or above, with the lower bound being 70% or greater, can seek this immediate change to a screening claim. So it's a point-of-care test, and they've done all the point-of-care studies and maybe already authorized. They can add a screening claim upon request by agreeing to do this serial testing program and doing the post-market study. Now, the existing templates for one-time use OTC and asymptomatic screening still are in existence. So if developers want to still use that pathway and get a one-time use claim, that's fine as well. But for the lower sensitivity, without data in the asymptomatic population, we want that serial testing as part of the authorization, and we want to confirm performance in a post-market study. Hopefully I addressed all of your questions. There were a few there.
Thank you. I was going to ask anything else. Are you going to release a template for the post-market study requirements in terms of sample size, number of positives, low positives?
So I think that's something that you can propose in a pre-EUA when you're requesting, or an EUA when you're requesting this, and we can confer with you. We expect that as the current template asks for 20 positives by a comparator, high-sensitivity molecular comparator method, that is still the expectation in the post-market study. So 20 positives should asymptomatic positives should do it.
Thank you.
Next question is from Paul Bartow. Your line is open.
Yes, good afternoon. How you doing, Tim? Quick question for you. So if we are contemplating being a lab and we are running an EUA authorized serology test, and let's say it's authorized on venipuncture blood. If we want to do testing using a dried blood spot, does that require a full validation, or is that just simply a comparison study with the existing sample method?
So let me just make sure I understand the question. So you have a serology test, and you want to have a sample type within your healthcare network that comes to your lab of a dried blood spot. Is this for a home collection or a healthcare collection?
I'd be interested in the answer on either one, but I was contemplating an at-home collection.
Yeah, so for at-home collection, that is not considered an LDT situation, but a device situation. And we have posted a template already with recommendations for validating a home dried blood spot collection. So I'd refer you that on our FDA website. If you are validating that as an additional sample type for your lab, for a kit that you're using that's authorized, you can do that within the confines of your healthcare system in healthcare environments, and it doesn't require an EUA submission. However, it would no longer be an EUA, considered an EUA authorized test.
Got it. That's helpful.
Okay.
Thank you.
Mm-hmm.
Next question is from Susan Sharp. Your line is open.
Thank you, Toby. Thank you, Tim. Quick question. If you collect a 10 swab pool with a single swab from each person, but you place it in the same transport tube or media, with the assumption that you will recollect the specimens from each one of those 10 people for individualized testing if that pool is positive, is that initial pool testing considered screening or surveillance?
That is a, can you say your name again? I didn't quite catch it.
Susan Sharp.
Oh, yes, Susan. Yeah, I mean, that is an on-the-edge question between surveillance and screening, and I would recommend that you come in with a question into the template email address with that question. Typically, when I've talked to people who are doing diagnostic or screening, looking at screening, they will collect 2 swabs at the time of collection and will retain the second swab in order to deconvolute that. Typically, diagnostic or screening testing provides results directly to an individual. If something's done purely for surveillance purposes, following all the FAQs and guidances that are provided by the FDA and CMS and CDC, then that sort of combined pool could be used in that kind of program. So it depends. It depends on the details. It depends on following those various FAQs and guidances on how to do screening testing or diagnostic testing and or surveillance testing. Whether you do purely surveillance or not, well, if you do purely surveillance, do also look at our template on how to validate this type of swab pooling, because it has different considerations than pooling media. In particular, with pooling media, you're more concerned about losing sensitivity to the dilution. When you're pooling swabs, the concern has to do with do you concentrate any inhibition in that swab pooling, or do you push the overall viral load in that sample above a threshold that is no longer detectable by that particular assay? So we've definitely seen, we projected it could be seen, and then we've now definitely seen that if you load up a pool, maybe with swabs with too high a viral load, that you can get a false negative on that pool, and that would be unfortunate. So I'm sorry I can't be a little bit more definitive, but it does depend, and happy to converse through the templates email box.
Okay, thank you. I did send in the question to the templates email, and the question, again, wasn't specifically answered, so I will try one more time. Thank you.
We'll also try to, if you do send it again, we'll alert the team to forward it to Toby. Actually, you can just ask them to forward it to Toby and Tim, okay?
Thank you.
Yeah, and I think we can also, clarify, and this is also in the FAQs, that, it does depend on what action you're taking. If the intent is to, use this to identify individuals that can or cannot take some action, whether it's return to school, return to work, whatever it is, that would generally fall under screening, not surveillance. But we're happy to discuss further for your particular situation as well.
Thank you.
Next question is from Kate Taylor. Your line is open.
Yeah, hi, Tim. I wanted to follow up on the earlier question from Jeff Andrews. So using this template, I understand that if we already have an authorized symptomatic antigen test with performance of greater than 80%, we would submit an amendment to our existing EUA to request a serial screening claim. And then we would propose in that intended use, whether that is, twice a week or 3 times, we would propose our intended use. And then with that negotiation, FDA would authorize, and we would then be obligated to do the post-market authorization study. Would that study be in asymptomatics? But would that then, if let's say we have twice a week serial testing screening claim, that post-market study would be done according to that. So you'd want to see, whatever you're trying to claim in the intended use, whether it's 2 or 3 times a week, you'd want to see that design in the post-market study. Did I understand that correctly?
That's absolutely correct. When it comes to OTC use, we're anticipating that most developers will choose to send instead of a single test, but 2 tests per package. Using this pathway, and they would instruct the user to test on day 1 and day 2, or day 1 and day 3, or day 1 and day 4, or something like that. And we would be looking for confirmation of whatever specific scheme you call for your serial testing is validated and shows adequate performance.
And this would be true whether you're a point of care device or an OTC device going for this, correct?
Correct.
That wouldn't change. Okay, thank you very much. That helps, Tim.
Next question is from Kodumba Venkamp. Your line is now open.
Good afternoon. Thank you for taking my call. Thanks for giving the new update about the screening and also about the Q serology template. So my question is related to the serology. Again, is there any screening test for serology that you are contemplating? What is the position of FDA on serology testing now that more than 70 million people got vaccination? So infection versus vaccination. So is there any guidance for serology? And in fact, there are many serology tests that have more than 95% on sensitivity, specificity, but they are all low priority, and that is not even being reviewed. We are adding more and more new screening tests. So if you can throw some light on that, I will appreciate. Thank you.
So when you're talking about a serology test in screening, its use could be considered for screening for donor selection for convalescent plasma donation. It could also be screening for people for measurement, say, of an immune response that may imply, with data supporting it, some sort of immunity or neutralizing antibody. Some developers talk to us about, can they use their serology tests to inform whether somebody has responded adequately in their adaptive immune response to a vaccine. I want to alert, though, this audience that we have started hearing reports that that some are using, some clinicians are ordering serology tests after vaccination, and some of those test results are coming back negative unexpectedly, and there is some concern. And when those investigations have been done, it turns out that vaccine generates antibodies to a spike protein, but the serology test is directed towards the N protein. So that serology test obviously is not going to be the ideal candidate for a clinician to order off-label a test to take a look at a response to vaccine. We remain open, as has been expressed before multiple times, to additional claims around serology tests. I think what I would say is we want to make sure and expand upon what I've just said about selecting the appropriate serology test to look for an immune response from a vaccine. And so we'll be looking at various pathways to do that and make sure that that's clear to laboratories and ordering clinicians. So, yes, again, we're open to these sort of things. We make decisions based on good science and data for a particular serology test and a particular indication. And as I just said, it may come down to the specific vaccine as well, whether a serology test functions for that specific vaccine. We also have stated that very high throughput tests of all categories are a priority, but low throughput, non-point of care, non-home tests are currently, and we don't envision making those a priority. We're looking to expand as fast as possible the available testing for COVID-19, from where it is now several-fold higher per day, and also expanding access. So that's going to be achieved with the higher throughput central lab tests, as well as access to point-of-care testing and home collection. So those categories still remain a high priority.
In fact, conventional immunology paired sample really can tell us about the recent infection. So just now how you are mentioning about the serial testing, based on the antibody to different time points, very clearly can indicate recent infection. Another thing is when you say the high throughput testing, where the serology is using high throughput, and what is the definition of high throughput where you need a serology high throughput testing?
So that is potentially a fluid target as the pandemic goes on. So if you have questions about whether your specific assay and assay format and workflow meet our current throughput expectations for review, just reach out either in a pre-EUA or to the template email address. For those tests that are already submitted, we do that review and prioritize accordingly.
Thank you.
Mm-hmm.
Next question is from Richard Montagna. Your line is now open.
Yes, this is Richard Montagna from Rionics. I've called in a few times, and thank you very much for the responses. They've all been very, very helpful. We have a PCR-based test that's authorized for use in either respiratory or saliva specimens, and we're now working to develop a saliva self-collection kit. And as you had recommended to us 2 weeks ago in a town hall meeting, we have submitted the pre-EUA to outline the usability studies. My question is, we see this being used in two different settings. One would be home collection, where the kit would be sent to a home user and they will collect the saliva and ship it back to the lab. And the second use would be what we're kind of calling mass collection, which kind of ties into your opening comments, screening at schools and places of employment, et cetera. So my question is, when we submit the EUA, can we lump both of those uses in a single EUA, and from an intended use standpoint, kind of lump them both together, or how would FDA see that working?
Yeah, so I think what you're talking about, so home use has a home collection. I would look to the home collection template, and that will give you some good indication for the validation studies for a home collection.
Yes.
I would encourage you to potentially use collection devices that have already been authorized for other assays, because most, if not all, of those vendors are willing to give developers write a reference letter that allows us to use their data for your submission, so you don't have to repeat a lot of the studies. At that point, it would be basically making sure that that collection device works with your particular assay.
Yeah, we were actually intending to use, we were intending to use the same collection device that we use when we had saliva authorized. So it would not be a new device coming in.
Okay. Well, that's fine. Just look at the additional home collection. It also would apply to remote collection other than the home that would be used for shipping, say, overnight, versus collecting and immediately transporting within your current time scheme. I think you're also talking about pooling saliva?
Not in this particular submission.
Okay.
We do have an EUA in there right now for pooling, but we're just talking.
So once you get,
go ahead.
Yeah, it also depends. I mean, if you want a screening claim, there are the recommended validations for screening claim, and you'd get an OTC. If you're to do a home prescription collection validation, that would work anywhere somebody writes a prescription for that collection method and your test, whether it be at schools or at home.
So this would be a sort of a generic prescription for a mass of people?
It would be a generic for, it's on the prescription, generic for whatever claims you come in with. If it's for symptomatic individuals alone, then if you've got all the stability, usability studies, then once we authorize, say, for home prescription use, that device could be used in any prescription setting other than the home as well.
Okay, thank you very much.
Mm-hmm.
Next question is from Shauna. Your line is now open.
Hi, thank you so much for taking my call today. I would like to ask you about the serial testing for screening purposes. My lab is SDI Labs. We are a CLIA lab, and we are working on a over-the-counter antigen test that would be then reflexed into a molecular test. So I just want to ask a few questions just to get confirmation of what was discussed in the last week's meeting. I understand that the FDA would like to know the demographics of the person who's taking the test and if the results came up positive or negative, because I understood that it is not a requirement for the person who's taking the test to register their results.
I want to clarify what your question is. You're talking about reporting requirements?
Yes. Reporting requirements for over-the-counter, antigen test.
Yeah, so if it's an over-the-counter test and it's performed at home outside of a laboratory, so there is no legal, federal legal requirement to report the results of that test. We are encouraging developers to provide an option for OTC users to report their results if they so choose. They cannot be compelled. we, in our authorization, do recommend that that patients report their results to their healthcare provider so that their healthcare provider is aware of that and potentially can help them out. So there is no requirement for pre-authorization requirement for a test to have a reporting feature for any type of test, let alone a home test. So I hope that clarifies that.
Yes, that does. I have a follow-on question, if you don't mind.
Okay.
So we would like, so with our over-the-counter test, antigen test, there would be an algorithm that would, determine whether this individual is highly likely to have COVID-19 or not, and then we would have them reflect it to a molecular test and send it back to us. What are your thoughts on it? What is the priority of your organization of FDA for such a test on the market?
So home tests are a priority. So you're talking about a piece of software that would be developed that would determine who gets tested in which form or way. And we are currently considering that a software as a medical device. And so the software would be reviewed as well as the testing program. And so you should look at some of the software requirements for such determination. And we will be looking particularly at the benefit risk of that kind. In particular, we don't necessarily mind that more people get reflex to a more sensitive test, but the risk is higher if you're eliminating people with your software who might actually be positive for SARS. And so that is a little bit riskier calculation. So if you want to come in with a pre-EUA for that sort of triaging software idea or whatever information you have on that, and ask us on what we think we can authorize around that, we would be open to it.
Thank you. And but just bear in mind that we do intend to send in 2 tests along with, for our over-the-counter. It won't be 1 test, and we would have asked them to take a test again within 3 days if we think that they are high risk or, they came up negative or they're high risk, and then send us back the same samples that they have collected for molecular testing.
Oh, okay. So, I think it depends. It's going to be the details here. It's a little bit hard on this call, but in some ways it sounds like you're mitigating risk with this software, and that would be a lower risk review. Okay, but I think it's the most important way is to give you definitive details on this program to submit a pre-EUA so that we can review your proposed plan and give you specific feedback.
Okay, thank you.
Next question is from Nisha Lee. Your line is now open.
Yep, thank you for taking my call. My question is regarding the use of retrospective specimen. I know you had reiterated the requirement of having the 10 to 20% of specimens being low positive to represent natural distribution of viral loads. I'm wondering if there are any requirements on moderate or high positive sample representation if, for example, having approximately 50% of the retrospective being high positive and maybe 35 being moderate, would that be something that's acceptable?
What we're aiming is to try to mimic the natural distribution of a virus, a CT, seen for whatever period of time after initiation of symptoms or for asymptomatics for the period for your program. We normally see, when a prospective trial, a very usual a very usual sort of spectrum of high to low positive samples. And when people use bank samples, we just want to try to reflect that. So there will be high positives, there will be low positives, and there will be moderate positives, and that's the primary consideration. I would, if you're having a method, we want to eliminate bias, so typically it's good to run your study design for using bank samples by the FDA through a pre-EUA submission to make sure that, depending on how you're doing it, that you're not introducing bias that would prevent us from making a positive regulatory decision. So typically when you use bank samples, we want to use consecutive samples collected from one date to another rather than sort of selecting individuals in between others. So that we see sort of that more normal distribution. I would also caution, and I've seen this happen, where a particular method of collection and types of bank samples that might be used, that it overemphasizes the low positives, and it does have a tendency to show a poor performance for the test, and we're not wanting to see that. So, be sure that the banked set of samples you're going to reflects a normal distribution, and then if you randomly select those from one point in time, calendar date to another calendar date, all the intervening samples, positives and negatives, you're going to eliminate bias, and you're going to help ensure that you don't have an overabundance of the low positives. And if you want a more detailed response than that, I would suggest submitting a pre-EUA to our team.
Okay, thank you very much. And just a quick question. I know you had mentioned that there's an update for the serology template, which includes variant testing. Will there be an update on the antigen template soon? And if our EUA studies are completed before that new variant testing requirement comes out, should we submit or should we obtain the variant testing data before submitting?
So, go ahead and submit now. We tend to, if someone submits something before we make a template update, we want to, as much as possible, adhere to our prior recommendations. That's the fair thing to do, unless there is clearly a high safety concern and we need to change that. But I don't see that happening right now. So we are revving the other templates, the other main templates, serology, molecular, and home. So be on the lookout for that, but I can't predict when they'll be posted.
Okay, thank you very much.
You can also take a look at the, the viral mutation guidance that we put out, which does signal the direction that we're heading. And then, as Tim said, we'll be updating the templates to reflect some of that as well. And if there is something that changes, it may be a post-authorization consideration as well.
Okay, thank you very much. Appreciate it.
Next question is from Johan. Your line is now open.
Yeah. Good evening. Can you hear me?
Yes, we can.
Hello? Okay, wonderful. I just wanted to come back on serology testing for monitoring the reaction of vaccinated individuals. Maybe I've missed it in some of the town halls, but did you actually already put up something like a guideline on numbers that one should test for maybe expanding the intended use claim for an already EU8 serological assay?
Yeah, so can you restate that question? I want to make sure I understand. I think you're taking an already EUA authorized serology test, and can you update the indications with additional submission and data.
Yeah, the question would be whether there are specific protocols already available that I could rely on in order to plan the experiments or the sample collections for the cohorts of vaccinated individuals in order to be able to. Hmm?
Yeah, go ahead, Vitters. Yeah.
In order to kind of build up a claim that I with this spike-based assay can measure the specific IgG reaction after vaccination.
Okay, so this would be an after vaccination. So we don't have any recommended authorization suggestions, study suggestions for that. So I would urge you to come in with the best plan you can come in with a probably a pre-EUA is the way to go, and suggest a study design that we can discuss with you.
Okay, and that would also be the way if the test is already submitted and confirmed by you.
Yes. I mean, that would be an additional claim for which we don't have current recommendations on how to validate the test for measuring the effectiveness of vaccinations.
Okay, good. Because it's clear, and you by choosing the correct assay with spike as an antigen, of course, in the lab-based situations we can see it that the reactivity is quite well measurable, but it's just a question on how to expand the claim that we did not have, not include.
I think there's different levels of claims. So, I mean, there can be one level of claim that says, oh, this antigen test, is able to detect antibodies raised by a certain vaccine. It's entirely different to say that those antibodies are protective against infection. So it all depends on your intended use claims about what sort of validations would be required.
Okay. Good. Okay, thank you. That was all.
All right, thank you.
And now I'd like to turn the call back over to Irene Ihir.
Thank you. This is Irene Ihir. We appreciate your participation and thoughtful questions during today's town hall. Today's presentation and transcript will be made available on the CDRH Learn webpage at www.fda.gov/training/CDRH Learn by Friday, March 27th. If you have additional questions about today's presentation, please email cdrh-EUA-templates@fda.hhs.gov. As we continue to hold these virtual town halls, we would appreciate your feedback. Following the conclusion of today's virtual town hall, please complete a short 13-question survey about your FDA CDRH virtual town hall experience. The survey can be found now on www.fda.gov/CDRH webinar. Again, thank you for participating, and this concludes today's virtual town hall.
This concludes today's call. Thank you for participation. You may disconnect at this time. Speakers, please stand by.